Now that we've taken a close look at the structure of the actin and the myosin inside the muscle, it's time to look at exactly how they work together to cause a muscle contraction. As I mentioned before, we use what's called the sliding filament model to describe what's happening inside a muscle as it contracts. We call it the sliding filament model because the filaments are going to slide against each other. We're going to have our thick filament of myosin, and it's going to pull on the actin, which is going to slide along it to shorten the sarcomere, which shortens the muscle cell and causes a muscle contraction. Let's start by looking at what's happening in a resting muscle cell. In a resting muscle cell, one that's not contracting, our actin filaments are covered by the tropomyosin. So remember, tropomyosin is that long, thin protein that's going to cover the active sites of the actin so that myosin can't bind to it. The tropomyosin is covering the active sites of actin because it's not bound to calcium. All of the calcium at this point is hiding in the sarcoplasmic reticulum of the muscle. No calcium is present. Tropomyosin is covering the active sites of actin. The myosin heads on the thick filaments are ready to start a contraction. They're in what I call the reach phase. Those myosin heads are reached out away from their tails and they are ready to go. They just don't have anything to bind to since the actin active sites are covered by the tropomyosin. In order to get our muscle contraction to start, we have to release the calcium. This is done by the action of a motor neuron. A motor neuron sends a signal to the muscle cell that results in the release of calcium from the sarcoplasmic reticulum. In a little while, we'll look a little more closely at how that happens, but for now, we're just going to say we have the signal from the muscle causes the release of calcium. The calcium then binds to the troponin on the tropomyosin. When the calcium binds to the troponin, it shifts shape, which moves the tropomyosin out of the way and uncovers the active sites of the actin. Now that the active sites on actin are uncovered, we can actually have a muscle contraction. In order to get the sliding filaments to slide, we have a series of 4 steps that are repeated over and over again. So remember that in our resting muscle, our myosin is reached out and ready to go. It's in a high energy state. As soon as the tropomyosin moves out of the way, our myosin is going to be able to grab onto the actin. That's the first step. I call it the grab, where the myosin binds to the active sites of actin. This is also called cross-bridge formation because we're forming a bridge between the thick filament and the thin filament when the myosin head binds to the actin. The second step is what I call the pull. In this case, the high energy myosin just bound to the actin, and it's going to return to its lower energy state. So this high energy reached out myosin moves to pull its head back towards its tail, and it pulls the actin along with it. That's also called the power stroke. That's an important point because that's where the movement of the filament is actually occurring, is during that pull or that power stroke. At this point, it's important to involve ATP. So now we've got the myosin returned to its low energy form. It's still bound to the actin. The myosin head is going to bind to a molecule of ATP. When myosin binds to ATP, it releases from the actin. That's what I call the release step. When the myosin head then hydrolyzes or breaks that ATP to use the energy, the myosin reaches back out and can then grab onto the actin again and pull. Then it binds to a new ATP to release, uses that ATP to take the high energy form, grabs the new actin, and pulls again. And that's what pulls the actin filament along the myosin filament, shortening the sarcomeres and causing a contraction. This cycle of steps repeats about 5 times per second, and it'll keep going until either the neuron stops signaling, meaning it's time to stop the contraction, or until the muscle cell runs out of ATP. If the muscle cell runs out of ATP, you can't release from the actin to go through the cycle again, so the contraction will stop there. If we take a look at what happens to the sarcomere as this contraction occurs, we can compare the sarcomere at the top of this figure to the sarcomere at the bottom of the figure. The top one is more relaxed, and the sarcomere at the bottom is more contracted. And you can see that we shorten the sarcomere. The distance between the Z discs gets shorter as we pull them in towards each other. So the sarcomere itself is going to get shorter. The length of the dark band, the A band, which is the length of the myosin filaments, doesn't change. The myosin filaments are staying exactly where they were. They're not changing in length. They're just pulling the actin along them. So our A band is going to remain the same. The I band, however, that area where we see just the actin without the myosin, will get shorter. As we pull those actin filaments across the myosin filaments, we have less area of actin that's not overlapped, so our I band is going to get smaller and smaller as the contraction continues. Once the muscle contraction is finished and we need to let the muscle relax, we pretty much have to undo all of the steps that we did to allow for the contraction. So the first thing that has to happen is the neuron has to stop signaling. Once the neuron has stopped signaling, we can start moving the calcium back into the sarcoplasmic reticulum. That means the calcium is going to let go of the troponin that's on the tropomyosin. When the calcium releases the troponin, then the tropomyosin covers the active sites on the actin, and once the active sites are covered, the myosin can no longer bind to it, and the sarcomere can relax. The relaxation gets a little bit of help from the titin protein. Remember that I described titin as sort of a big spring holding those thick filaments in place. As soon as the myosin is no longer holding onto the actin, those titin springs can push the Z discs apart and the muscle can return to its original length. This is often also helped by the action of opposing muscles. So let's say I contracted my biceps brachii muscle in order to flex my forearm. That's contracting the biceps. When I relax that contraction, I can use action from the triceps muscle to then help stretch my biceps muscle back to its original length, in addition to the action of the titin and other structures inside the muscle.